Science in Pajamas. Alright, you guys, welcome back. Today we're going to talk about cell transport, so how things move in and out of the cell. Specifically, we're going to be looking at something called passive transport. Now, passive transport is when things can move across the cell membrane without expending any energy. And the reason why certain molecules are able to do that is because of something called concentration gradient. Now, concentration is the amount of a substance or particles of a solute in a given amount. So it's kind of like the density of it. Now, I'm going to give you guys an example. Let's say there are two classrooms right across the hall from each other. All right? So we have classroom 1 and classroom 2. In classroom 1, we have a lot of kids. Not good in the time of Corona. As you can see, we have a lot of kids in here. We have 15 kids. That's how many I drew. It may not sound like a lot, but in terms of the space, look at that. And in classroom 2, we only have 5 kids. So here we have 15 kids. Here we have 5 kids. What do you think the kids would want to do? Do you think they would want to stay all cramped up in this classroom? Do you think that some of the kids from class 2 would want to move into this classroom? Or do you think some of the kids from classroom 1 will want to move into the less crowded classroom 2? Well, the answer is the last one. Some of the kids will want to move from the higher concentration to the lower concentration of students. And that's what passive transport is. The crowding of the molecules essentially forces them to move down the concentration gradient from high to low. So that what happens is we would end up with So what happens now is we've reached equilibrium. Equilibrium is when the concentration is equal on both sides. Now here's the interesting thing about equilibrium. Just because you've reached equilibrium doesn't mean that movement between the two classes stops. What it means is that movement occurs in both directions equally so that each side will maintain about an equal amount. So that is what we call equilibrium. Again, particles keep moving, or in this case, the kids keep moving back and forth, but we end up with about the same on each room. So maybe 2 move over there, and 2 would come back. 3 would move over there, 3 would come back. 1 would move over there, 1 would come back. So it stays roughly equal. Now in passive transport, things do that all on their own. The particles are going to move across all on their own usually. Now, let's see. I'm going to make a membrane. This one's nice and fluffy. Sorry, I'm trying to look for some things I can use. Alright, so these are going to be my memory. Now remember, it's a bilayer. Let me get some more things. Ones that match or roughly match. Dun 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 dun. Okay, so we got this and this. I have a whole bunch of pipe cleaners because they're useful for stuff like this. So bear with me while I look for some that match. Oh, interesting shapes. All right. So these will work the best. Alrighty. So I'm going to show you what we're talking about. You can see that all right? Yes. Where'd my magnets go? Alrighty, so we have our membrane. Now what that means is that on either side, it's like that. This would be the outside. Is that showing up all right? No, it's not. All right. So this would be the outside of the cell. And this would be the inside. So far, so good? All right. Now with diffusion, what that means is that we have certain molecules that maybe are small enough they can just kind of sneak right on through the membrane. So they can actually go right across it. So we have some molecules hanging out here. Now if you notice, there's a higher concentration of these little yellow ones outside of the cell than there are inside. So what's going to happen is the higher concentration is going to cause this one to move right on through the membrane and into the cell. I'm just going to keep going. Until it's equal. Now we've reached equilibrium. Now in the case of equilibrium, again, that doesn't mean that movement stops. It just means that it continues, but in both directions. So that each side remains roughly the same. So that's what diffusion is. However, sometimes molecules might be too big to cross the membrane. They might be too big. It can't get through on its own. So what happens instead, this is still something that the cell wants inside of it so that it can use it for materials to make more proteins or to make organelles or whatever. Or maybe it's glucose. It needs glucose in it so that it can, power the mitochondria. But these are too big, or maybe they have too much of a charge on them. So in which case, we have something called facilitated diffusion. Now what facilitated diffusion is sometimes there are these things called, I'm trying to get some pipe cleaners out, channels. They're protein channels. Well, if they're a protein channel, anyone want to guess what they're made out of? If you guess proteins, you are correct. You would be correct. So what a protein channel is, it is proteins that have kind of come together to make an opening, almost like a tunnel. So there we go. We got our little tunnel. I get our membrane back up on this side now. So now we have this protein tunnel. Now what it does is it'll allow these larger molecules to pass through. But these channels are very, very, very particular. This will still keep going down the concentration gradient. However, if something else tried to go through it, it actually wouldn't be able to. They're very, very highly specific. These proteins won't allow anything other than this guy to go through. So that won't be able to go through, that won't be able to go through, only these green ones. Now, we call this facilitated diffusion because it's diffusion. It's going from concentration of high to low, but it needs some help, and that's where these protein channels come in. All right. Well, we also know that diffusion of water, we call that osmosis. All that is, it's a fancy term for when we talk about just water. I got my mini bins of magnets, bear with me. So water is very, very important to the cell. It keeps it hydrated, it allows other reactions to occur. So we need water. We do. Now water is small enough that it can actually just go through the membrane. So it's diffusing from high to low concentration once again. We just give it a special term, osmosis, because we do consider it pretty special. All right. We're going to get rid of some of these guys. There are some cases where we have to balance. Remember, the goal is to reach equilibrium. Equilibrium actually requires less energy and it's more stable for the cell. So the point is to reach equilibrium. Close up that. Cells won't be able to go through. Let's see. Now, if you notice, I am making a situation where things are unequal. All right, so all the big ones, doesn't matter their color, all the big ones represent a larger solute. So it's a larger solute. And it doesn't matter what their color is. And then all the small ones, once again, regardless of color, all the small ones represent water. All right, is that all the small ones I have? Oh, no, got some down here. So you'll notice that the concentration of the solute on the outside of the cell is different than the concentration of the solute on the inside. Now the water is at equilibrium by itself. There are 9 molecules of water here, 9 molecules of water there. But the solute itself is not at equilibrium. Now this creates something called osmotic pressure. These molecules are too big. They can't diffuse across and they can't go through the channels. So they can't diffuse across on their own in order to reach equilibrium. So what has to happen instead is if you can't move the solutes across the membrane, you move the water. Because remember we said that concentration is kind of like density. So we're looking for the amount of solutes per volume of the solution, which is the water. So if you can't change the amount of solute, you can change the amount of the solution. So we have more of these molecules out here than we have in here. What we want to do is we want to dilute the outside. We want to try and make it so there's a larger volume of water in order to dilute this higher concentration of the solutes. So water is actually going to leave the cell. It's going to be pulled out of the cell by something called osmotic pressure. Now, it doesn't mean all the water would necessarily leave, but a good amount will in order to try and dilute the high concentration on the outside. So when you have a higher concentration of solute in the solution outside the cell, we call that a hypertonic solution. And notice we have very little water inside the cell now. So what would happen with this, so we said this is hypertonic. This is a hypertonic solution. You have a higher concentration of solute outside of the cell, so the water gets pulled out in order to dilute it down. That's going to cause less water inside the cell. It's going to become dehydrated, and it can actually shrivel up and die. All right. Let's see. 5, 6, 7, 8, 9. Alright, well now we have the opposite scenario. Now we have a higher concentration of solute inside the cell than we do outside. Again, the water's starting out at equilibrium, but these large particles, they can't cross the membrane or go through the channels in order to reach equilibrium for the solute. So just like before, if we can't reach equilibrium by moving the solute, we will reach equilibrium by moving the water. So what we have to do now is, since inside has a higher concentration, we want to move more water into the cell. This will help to dilute the particles, the solute, because now they have more solution that they are sitting in. Now if you notice, getting a little bit crowded inside the cell, isn't it? We have all this water moving in. We call this a hypotonic. Now review these once we're done. Again, it's the osmotic pressure. Because we can't change the concentration of the solutes on their own, we move the water in order to create more liquid inside the cell to dilute the higher concentration of the particles. Now, since all this water is going in now, it's actually going to blow up like a balloon. So it's going to get rounder and fatter. And if it keeps taking in more and more water, it can actually burst. All right. last one. Let's start with these. 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So it's 18. So 1, 2, 3, 4. All right. Now here we have equilibrium already. So we have an equal concentration of the solutes on the outside of the cell as well as the inside of the cell. Now once again, water is still going to move. Diffusion of the water is still going to occur. But since everything is at equilibrium, we're going to see them move in both directions to help maintain that equilibrium. So maybe this guy moves over here. This guy moves there. So water is still going to go back and forth, but it's going to do so equally. Now, since everything is already equal, we call this an isotonic solution. And that just means that the concentration of the solutes are equal outside the cell as well as inside. So yes, water will move across the membrane. It'll go through osmosis, but it'll do so equally in both directions. All right, I do want us to kind of review what will happen with those three, looking at them all together. Whee, 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 whee. That was a little too much fun, huh? All right, so in conclusion for those, when we have a solution, when the cell, let's see, I'm just going to start off with 3 cells. And our all right, so here we have the 3 situations. Now, first one we said was hypertonic. So it's a hypertonic solution. There is a greater concentration. We have a greater concentration of the solute outside the cell than inside. This is going to cause the water to be pulled out of the cell in order to try and dilute the surrounding solution, which will then cause that cell to wither. It's going to become dehydrated and it's going to wither. Here we have isotonic. And in this case, the concentration of the solute is equal both inside and outside the cell. So that means the cell is sitting in an isotonic solution. Yes, there will be movement of water, but it'll move in both ways equally to keep everything in equilibrium. And then here we have hypotonic. And this is when there's a greater concentration of the solutes inside the cell. As a result, and again, if we can't move the solutes, we're going to cause the water to flow into the cell in order to dilute the concentration inside the cell. This is going to cause the cell to get really, really big, because like a water balloon. When you fill a balloon with water, it gets really, 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 really big, and then it might even burst. So just like if you put too much water in a balloon, yes, you start off with a small balloon, you put in the water, it gets bigger, but if you keep filling it up and keep filling it up and keep filling it up, eventually it can't hold anymore and it'll break, it'll burst open. And that's what could happen in a hypotonic solution. All right, so I hope this helped. Hopefully, if you have any other questions, you'll come see me, class, email, etc. But I hope it did help to kind of explain a little bit about passive transport. So in the meantime, you guys, take care of yourselves, stay awesome and amazing, and I will see you next time. All right, you guys, take care. Bye-bye.